So welcome everybody to the latest Science for ME video. This time a bit different from what we've done previously. We're fortunate enough to be discussing the latest 2019 Ramsay Grant Programme Awards with Alison Sadie from Solve MECFS. So welcome.
Hi, thanks Andy.
And to kick us off, could you perhaps basically just give an overview of what the Ramsay Grant Programme is and what it functions, what it's there to do?
Sure, yeah. So it's a grant programme where we offer small one-year grants to investigators to attract people into the MECFS field, keep them engaged, allow them to generate seed data that can be used for larger grants, NIH grants or other foundation grants. So we really view it as our sort of pipeline to build an MECFS research workforce. It's now in its fourth year, I believe. Mm-hmm. And you know, it continues to gain momentum. So we're pretty excited about the traction that we got this year.
So to give our viewers a bit of an idea, like you say, that they're intended to be seed grants. Mm-hmm. So it's not the unfortunately multi-millions that we might hope to see spent on research programs for MECFS. So it's smaller to allow data to be gathered and then the idea is that it's built on from there. Exactly
that. So they're $35,000 to about $50,000 approximately. That's the range for a year. And then over the past 18 months, we've started to sort of embed some things that help them get to that next level of grants. So we're offering grant writing support to the Ramsay awardees so that we can not just passively, you know, expect them to apply for grants, that we can actively support that process. And then we're also doing a number of things to connect people together so that there is that sense of community. So that people feel engaged, they want to stay in this field, they can share methods, samples, information, learnings, and then ultimately we're hoping that we'll be able to potentially get people together to collaborate on large NIH grant proposals.
Okay. And this year, the number of applications, were you able to increase those? Did you see those increase?
Yeah, so we did a couple of things that were new for this year. So Alison put together an amazing researcher toolkit, and we can share a link when this video goes out. It's a really great resource that outlines all of the areas of science which are of interest. It talks about disease models. It gives everybody, or it gives a researcher all the information they would need to start to think about a research proposal. And we did that primarily because a big goal of the program is to bring in people who are new to the field. So someone, for example, who might be researching immunology in cancer, we wanted to try and attract those kind of people and give them the toolkit to be able to quickly understand the disease. So we did, that was one thing. And then we did really wide outreach. So we posted the request for,
Request for
proposals on like lots of, you know, other academic websites. We posted it on a website which aggregates lots of different universities. We partnered with the NIH, they pushed it out through their channels. So we tried to get a really wide distribution of the announcement in the hopes that we could attract additional people to apply. And we were thrilled. We had, I think, 15 in total and 12 that advanced into the peer review process.
And in addition, we also had, I think, at least 4 or 5 investigators who ultimately couldn't make the deadline for this request for proposals, but that are really interested and highly motivated to apply for 2020. So we already have some in the pipeline.
Yeah, and one thing we've kind of learned through this process is that we feel like we need to have a longer lead time. So we're thinking even for next year we might open the cycle even earlier and give people enough time and try and increase those numbers even more.
Great. So you were kind enough to give me a bit of a head start look at the proposals. Just in broad themes first, before we look at the individual projects funded. was there a conscious decision to try and make it as diverse as possible? Because you funded 7 projects and there's 6, I would say, broad themes to the projects funded. Was that conscious, or was that just, you know, how it fell out really?
No, so we do make a conscious decision to have a variety of different proposals focusing on different areas of science. And the reason we do that is because we don't believe that there's any one path which has presented itself as the silver bullet of the cause, or you know, a potential treatment target for this disease. And so we think that there is value in continuing to be broad and interrogating lots of different areas of science. One thing we did do new for this year, we did have a carve out for a collaborative proposal. So that was a proposal where we had at least 2 investigators working together. And I think we had at least 3 of the proposals that we funded actually fell into that collaborative carve out. And we're thrilled about that.
Yeah, and it was also across institutions. So it had to be 2 investigators from 2 different labs or institutions. And so we were thrilled to get 3, and we were able to fund all.
And they were all highly rated, so we were thrilled with that.
Brilliant. One thing that just before we get into the individual discussions of the projects, so the selection criteria for patients, for samples, that you insist as PEM as being you know, a defining thing, that, you know, any samples and patients involved, you know, have to report having PEM. Is that, am I right in thinking that? Yes,
absolutely. So in the toolkit that Sadie mentioned, you know, we didn't specify a certain case criteria for participation, but we did stipulate that in order for someone to qualify as having MECFS, then that PEM should be indicated. And the majority of the proposals did include research case definitions that fulfilled that, and the few that didn't was flagged both by us and by our peer reviewers, and the investigators were required to integrate a case definition that included PEM into the proposal to be funded. So, and all did.
Okay, brilliant. Right, so kicking off with the first one, nice short title compared to some of the others. So
I have another version of that title if you want the longer one. We'll leave it at that.
So the first one is entitled Altered T cells in MECFS. So T cells being part of the immune system. So I've got that far. T cells have come up as a target before. So does this project differ in any way to some of the kind of avenues followed or discussed?
Yeah, I mean, as you mentioned, T cells are kind of a hot area of late. The last, couple years, Daria Unatmatz at JAX, for example, has been doing work in this area. He was kind of one of the first ones to propose it. This proposal kind of dovetails with that work, but it also identifies a sort of new subset of T cells, so like a sort of subgroup of these T cells that could be driving the dysfunction in the immune systems of people with MECFS. And so this investigator, Lisa Sellin, she's a highly experienced investigator working with Anna Gill, and they're new to MECFS. They did do some preliminary, very, very small pilot work that informed their Ramsay application, but this would be really expanding on that. And, if they're successful, it could really identify either an infectious trigger that is influencing these T cell populations, or an autoimmunity sort of pathway. And so it does build on some previous work and would be consistent with some of it, but it does have sort of a novel aspect to it, and they are new investigators, which is very cool.
Yeah, I mean, the other thing to note about this proposal is this was our highest ever scored proposal in the history of the program. We rank them on a NIH measure, which is 1 to 10, with 1 being the highest, 10 being the lowest, and this came in at a 1.5, so we were thrilled.
Wonderful. Which isn't to say I would imagine that the rest of them were particularly low scoring at all. Just
no. They were all high scoring, but this one particularly stood out. Yeah.
Fantastic. So next one slightly more, I say slightly more complex, which suggests perhaps that Tcells isn't complex, but which obviously isn't the case. But so the title for this one is Possible Class 2 MHC, which I'll leave that for you to pronounce. MHC deficiency in patients. So major histocompatibility.
Mm-hmm. Yeah, another
MHC. So, so it's not something that I've seen come up on the forum or talked about that particular phrase. So, but from my first just brief look that I've been able to in the time that I've had the details, MHC, I would characterize it, whether it's right or not, as kind of a base to immune cells. It's a point before immune cells are in action, or it's some kind of precursor to them.
Like a pathway, yeah.
So you know, really what attracted you to that project? You know, what made it interesting enough to fund, really, is my question.
Yeah, so this was another proposal that was attracted to us because it had a really highly specific testable hypothesis that if the investigators are able to confirm it, it could have it could explain the symptoms for at least a subset of people. And if they can't confirm it, then that's really useful information because it gives us okay, we've kind of explored that potential here, and it could, inform research going forward in that way. That's another aspect of the Ramsay that we think is really important, is that negative findings are also very, very informative. And
right.
And so that was one aspect of the project that we liked, that it was really specific and highly testable. Then, peer reviewers that reviewed it both had, one had a lot of expertise in inheritable factors, and the other had a lot of expertise in immunology, and they were both highly enthusiastic about the potential for major histocompatibility deficiency to explain the symptoms for some people. And so I kind of deferred to their expertise in as far as the nuts and bolts of the science, but for me, I found it to be very compelling, and even if it's something that can't be confirmed, it would be really highly informative as well.
It was, again, highly scored. It was very well regarded by our reviewers.
And it's nice to see that I believe the lead investigator is based in Spain, or indeed him and his team.
Yes.
So
yeah, I think half of our proposals are US based and half are in Europe. So we quite like that, I think.
Yeah.
Distribution.
I mean, I think the challenge is, obviously we know the US system, so we know the NIH path, and we want to also encourage our European Ramsay investigators to find their equivalent of the NIH path. And as I mentioned before, we're going to be very proactive in supporting that process.
Brilliant. And am I right in thinking that this particular project is one of those collaborative ones? Have I got that right?
No, it's actually not. Yeah, this one is all with investigators at the University of Navarra.
Ah, okay.
But we'll be sure to flag the collaborative ones for you.
The next one coming up is a collaborative one.
Yes.
How about that?
Okay. All right, brilliant.
But this project did include, I think it's 6 investigators, so a huge amount. The Bruno Peva runs, I think it's the flow cytometry core, and so he's brought in this whole really highly experienced, cool team, and they're working also with a med student as well. So in that sense, it's very collaborative.
And new to the field, I believe. Is that right?
Mm-hmm.
Yeah. Which obviously is something that we're all very happy to see. The more scientists that we can investigate interest in investigating ME, hopefully the better. So quite a subject change from the immune system for the previous two to this next project, which is entitled Defining the Postural Contributors to PEM. involving a wearable sensor device, I believe the description says. So it's aiming to give a kind of measurable or a means to measure post-exertional malaise.
Mm-hmm.
Is that right? Yeah.
Yes, yeah. So Shad Roundy, who's the investigator here, also new to the field, is an engineer at the University of Utah. And he wants to validate a wearable sensor that's worn on both ankles that would basically establish an algorithm to measure both severity and PEM, be like sort of like a clinical endpoint for those things. And a lot of wearables are calibrated for individuals that don't have something like a disease like MECFS. And so that's a really strong aspect, is that when you incorporate wearables into a study, you really want them to work in the population that you're studying. And this is another one of the collaborative ones that Sadie might speak to a bit.
Mm-hmm. Yeah, I mean, I think this was really compelling for a couple of reasons. It was a collaborative proposal. It had a really strong clinical leaning. It's in partnership with Cindy Bateman and the Bateman Hearing Center. And then they had a significant amount of early data that they used kind of in their application. And I think there's high potential for this one to go on quite easily to a bigger NIH grant. So it had a number of factors which made it very compelling, but one of them was certainly the collaborative aspect to it.
And presumably, if it's successful, it could be something that relatively easily is then usable in almost any other MECFS project that there might be out there.
Mm-hmm. Very much.
Yes, it has applicability in that sense. And I mean, Cindy, she does so much great research on the clinical side and has so much information about her patient population that that collaboration, I think, is going to lead to great results.
Brilliant. Okay, bit of a longer title, this one. So we're moving on to one that looks for evidence of persistent pathogens, I think would be the correct characterization. Well, actually, the title being Extensive Characterization of the MECFS blood and CSF, cerebral spinal fluid, presumably, microbiome and virome. So in very basic layman's terms, they're looking for bugs in us, I guess.
Yes. Yeah, and this is another one of those collaborative proposals. So this has multiple institutions working together. And I'll pass it on to Alison to talk about the project.
Yeah, so it is highly collaborative. I think there's 3 institutions that are involved. There's actually really, there's like 4, I think, institutions, but the main kind of principal investigators are Nikos Kiripidis and David Paez-Espino. I know I'm trying to get the pronunciation. They're affiliated with the Joint Genome Institute at Berkeley. There's also Chris Forbes, as well as a previous Ramsay awardee, Jonas Bergquist, who is he's spoken a few times about doing things with cerebrospinal fluid, and so he's really bringing a lot of punch with that, and it's always really cool to see a previous Ramsay investigator involved in a new project like that as a collaborator. Additionally, there's Robert Moore from Harvard, and so it's really just extremely collaborative. He leads a lot of studies looking at how persistent pathogens in human brain tissue can drive neuroinflammation. And so it knits together with some other work in related conditions. Right, it's just really interesting. It's kind of I think a proposal that will be really valuable because the gut microbiome is just really buzzy in general. It's been of interest in the MECFS field, but it's really been very limited to bugs in the gut, right? And mostly bacterial populations, because those databases are the most well validated, essentially. And so this will open up studies of viruses and other things. So we're looking forward to what the output from this could be. It could really open up some new lines of investigation.
So presumably with the involvement of Jonas Bergquist, it's a truly international project as well. So
I think it makes for it's the 4th international collaborative that we've funded, maybe third or fourth over the course of the entirety of Ramsay.
Wonderful. Next one that is, well, another European, I believe, Dr. Francisco Westermeyer, is based in Austria. And he'll be looking at unraveling the endothelial function in MECFS. So basically looking at cardiovascular function. Yeah. I mean, can you tell us more about that? Is, will he be working by himself? I can see he's got experience in diabetes and other areas, particularly cardiovascular. So I think in your description you mentioned that's a collaborative group as well. So
it is, yeah. That's another one of the collaborative ones. And are they collaborating in country, or is this another transatlantic?
It's a trans-European collaborative. So it's with another investigator at the London School of Hygiene and Tropical Medicine, another longer title. But yeah, Dr. Francisco Westermeyer, I do believe he's involved in Euromine a bit, which is that European ME network of researchers, which is really great because, we've funded in the past a Euromine researcher, Carmen Scheibenbogen, so it's great to fund another that's part of a network that's, been building in Europe as well, so it can kind of tap into that. Yeah, this one is really interesting. Endothelial dysfunction is kind of interest because there are so many reports of vascular abnormalities in people with ME. And it's previously been not really studied, and he has so much experience that he can really do a deep clinical investigation of people. He's done some in vitro studies, so in the lab. He did some very preliminary work, but this would be an in vivo study in the body. And so they'll do a really deep clinical characterization of endothelial dysfunction, markers of it in people with ME, and then they'll also take blood and look at inflammatory markers that are particularly associated with endothelial dysfunction. So it's kind of really, it's quite novel, I think. And again, it's really very interesting and could have a lot of explanatory power for at least a subset of people.
I think possibly to explain to our viewers, endothelial cells, am I right in thinking that they're basically the cells that are on the inside of blood vessels? Is that correct?
Yeah, that's right.
So, many patients have reported issues with, blood pressure and such like. So presumably this is looking at that kind of area, and obviously problems with blood pressure then has effects on heart and all the organs in the body. So.
Yeah, and exactly
that, yeah.
Okay, so moving on to the next, passing post-exertional malaise. So title continues, does post-exertional autonomic recovery impact post-exertional malaise? Other than that it looks at the autonomic nervous system, I was wondering if you could explain a bit more with this one. So this one's a US-based project, I believe.
Yeah, and I'll just give a quick shout out to the lead investigator, Keegan Monaghetti, who obviously came up with a very clever title with the parsing. It's like when people, in the United States Congress, they like to come up with acronyms that spell out what the bill is about. And so I bet he did that and was like, nailed it. So good. It was a very good title. So he's a Stanford cardiopulmonologist, and he has published in MECFS before. He did a study that used an exercise test to kind of look at inflammatory markers in the blood. That was one of the things that we outlined in our toolkit is of high interest, and it's of interest in the field generally, but we really wanted Ramsay's to apply with these sort of provocational studies where obviously, within MECFS patients, they experience PEM, and that's provoked by something. And you kind of want to understand what the body physiologically is like when they're in that state of PEM. And it's a lot to put patients through, but it's also really highly meaningful for studies. And so he has some experience in administering exercise tests within a study. And so within this study, he'll be looking to understand the potential connection between dysfunction in the autonomic nervous system and PEM. And so they will ask patients and healthy controls to get on an exercise bike. They'll measure them at baseline, so before exerting themselves. They'll do some symptom self-report. They'll try and look at their heart rate and heart rate variability, which is also something of really big interest in the field. A lot of patients report having kind of abnormal heart rate variability. And then throughout the course of the exercise test, they will measure cognitive function, return to baseline, so how fast the heart rate returns. So they're going to assess for different measures of kind of autonomic function. They'll also, through the question, the questions that they administer to patients, they'll ask about, try to get at PEM as well and try and associate these two. And that's kind of novel. That's not really been directly investigated before. So that's kind of a big interest. In addition, I do think the investigators are going to do follow-up time points electronically. So, PEM for some people can range out quite to, a week, couple weeks. And so they'll also be administering questionnaires at longer time points as well. Sorry, I feel like that was a bit long.
No, that was great.
That's actually great to hear that they're looking at longer time frames, because many, or at least of the ones that come to my mind, many studies that have included PEM as something that's looked at, it can often simply be they look at the results from up to 48 hours after the exertional event, and while that may capture quite a large proportion of patients, like you say, many patients have symptoms that extend potentially far beyond that period. So certainly I think something that looks at a longer time range is very useful.
So I think they are looking early, but they're also looking over time. So I think that's really going to help when they're trying to do these associations between the autonomic nervous system and the incidence of PEM. So we're excited to see what comes out of it, and a great group of researchers.
Yeah, he's teamed up with Julia Leib, and she's a young investigator who's been involved in ME research, and it's really cool to see a continuation of her involvement, as well as Dr. Lily Chu, who's pretty influential, very influential. So it's a great group. And
finally, we've got a project that is looking at brain perfusion. Funnily enough, with someone that I spoke to just a couple of weeks ago, who was talking about brain perfusion. So, so Dr. Mike Van Elzakker is looking at brain perfusion changes in CFS before and after exercise challenge. So again, something else that he's looking at PEM and the effects of it, but this time in the brain. And for those out there who are not too sure about brain perfusion, basically, as I understand it, it's about blood flow in the brain and what changes there may or may not be. So he's got a number of collaborators on that as well, I believe.
He does, yeah. He has Dr. Kenneth Kwan, who was literally one of the investigators who helped invent the fMRI and validate it, which is very cool, who previously I don't think has engaged in studying MECFS populations, as well as Dr. Chan, who has a lot of expertise with fMRI and specifically how you can measure, I think, blood flow using it. And so there's a lot of deep expertise there from two people that have previously not been involved in the field. As well as Mike Van Elzakker of brainstem fame, famously. He's been very influential in thinking about how we can use better methodology in assessing some of the neurological abnormalities. And so I think it's a really cool.
It's a great collaboration, and again, it's close to the clinical side of things, so it's compelling from that perspective that we can immediately get some clinical data. It'll be interesting to see how this part of it doesn't align with Jared's work, right, in the brain and his imaging. So we think it's going to be really interesting.
And in addition, you know, speaking about longitudinal time points, he's doing a cross-sectional study where they're looking at one time point, but they have a deep interest in future study where they can look at different time points. And so this could help set them up to do that kind of longitudinal research study.
Okay, brilliant. So do either of you have a particular favorite project out there?
Oh, pick a favorite.
Oh, I sure secretly you must do. Well,
I always like the ones that are a little more clinical, right? That's been the most of my career has been in the clinical space, so I'm always a little bit more excited about some of the clinical ones. But I think just in general, we're thrilled with the types of projects, the novelty of the projects that should come through, the collaborative elements, and you know, people really seem to take that to heart, and you see that, right, in that 3 of the proposals are collaborative proposals, and then that we have new people and young investigators brought into the program, which is another key goal for us. So I know that doesn't answer your question, but we can't really.
Well, it was slightly a tongue-in-cheek question, really. So I'll accept that answer for now. And something that I intended mentioning at the start, but the key thing that, or a key point that needs to be made, is that you can only make these grants through donations, essentially.
Yeah.
But without those donations, that, you know, if you don't have those donations, then you're not able to make these grants to encourage investigators to look at MECFS, you know, and to build up what you're looking to build up. So on your behalf, I'd make a request, you know, for everybody watching and listening, if it's at all possible, consider donating to Solve MECFS.
We'd really appreciate that, and even small donations, right, can add up, and collectively it can be enough to fund a proposal. So you're absolutely right, this whole program is funded by our donor base, and you know, the more money we have, the more proposals we can fund, and the more we can grow this program and really truly build that MECFS research workforce.
Excellent. And to put you on the spot, any last thoughts? I mean, you kind of covered, you know, you gave a hint about which project interests you the most, but then complimented them all. But
I think you know, we're excited. We're excited to see what comes out of it. As I mentioned, we really want to be more proactive in the bigger grant writing process. The other thing that we're excited about is we're hoping to do a Ramsay conference in the first part of 2020, where we're bringing together not just the 2019 class, but all the previous classes, as many as are able to attend, to share findings, methodologies, positive, negative results. But there's also going to be a part of that program which is actually workshop based, where people can sit down together and start to map out the outline of a collaborative grant proposal to NIH. So we're pretty excited about that, both from an engagement standpoint, but also from a progressing this early data into something that's more significant.
Okay, wonderful. And just to make the point again, you'll be looking to kick off next year's Ramsay or applications for the Ramsay Awards earlier, January time, I believe.
Yeah, I think we think so. When did we do it this year?
Gosh,
May,
I think
April?
Yeah. We started the drumbeat like end of March, and then April was really. So I mean, now we're kind of on the record as saying January.
At first, we're going to open it in January, probably.
Well, yes.
But we definitely want to give more lead time. I think, I mean, it's just, if you don't work in this field, it's just going to take you longer to put a proposal together. That's just the fact of the matter. And while we did a great job with this extended outreach, it would have been even more productive had we had more time to explore more avenues and push even more. So I think just giving ourselves that runway, there's no reason why not. and so we're certainly going to give more runway this year.
So to end, for patients, consider donating. And for any researchers watching, get your thinking caps on and get ready for the application window to open.
And make sure to email research@solvecfs.org, any researchers. Would love to add you to my folder of prospective Ramsay 2020.
Yeah, and we're also happy to work with people. You know, if someone has the seed of an idea and they don't quite know how to turn it into a proposal for ME, we'd be more than happy to work with them. So, you know, that door is always open.
Wonderful. Right, we'll finish it there, I believe. Nice quick overview of your projects, or the projects that you funded. So thank you both for your time.
Yeah, no problem. Thank you, Andy. We really appreciate it. And hopefully the sneak peek hasn't been too dry. I know sometimes when we're talking about the research, it can be a bit difficult to sort of stay engaged. So hopefully everyone made it through to the end of us.
Well, knowing you're the Science for ME base, they'll probably be wanting more detail. So we'll just refer them to Mike Van Elzakker's interview with you and other sources.
Yeah, our forum members no doubt would love to have all the intimate details of the projects. But yeah, this was intended as a brief overview for those people out there who would appreciate a bit more than, you know, obviously anything that you can give in a normal press release, that kind of thing. So again, thank you very much for your time, and fingers crossed we'll be able to speak soon.
Yeah. Thank you, Andy.
Thanks.
Bye for now.